Okay, we are live to talk about fins to limbs, mostly tetrapods, and a little bit on vertebrate paleontology, which is kind of pissing me off, to tell you the truth.
Wait, why?
Oh, vertebrates just piss me off in general. I
Fair
very
enough.
seldom get mad at worms.
That is true. Yeah, I can't remember the last time I got mad at like a spiralian.
Yeah, it's mostly vertebrates that piss me off. But no, the looking over phylogenies and trying to get a handle on the overview of the phylogeny of vertebrates, for instance, or chordata, it's a little difficult sometimes because some of the diagrams are all over the place. And
How so?
well, you almost need a giant room to paste everything together to get an overall picture of things.
For sure.
Yeah, I do have some pictures that I snapped that we can take a look at some point.
I want to, like, whenever I become financially independent, I want to get like one of those huge phylogeny of all of life, like the big circles that you see. Just get a huge like 6 foot by 6 foot one and just put it on a wall.
Yes. Yeah, and I'd like to get one of the geological time periods and and put that on the wall. I've been trying to find a good one so that I can buy, because I will find a wall to put it on. I don't have many walls left, but I will make a wall if I have to.
I mean, you'll get the bricks out just for this.
I could hang it in front of one of my whiteboards. It'd be really cool if it was like a shade that I could pull down. Like, shade, zip, that sort of thing.
That'd have to be like custom made, probably.
They have, I don't know, they used to have those all the time in the classroom where you could pull down the shades. There'd be a chart on it, right? So there must be something like that. But if you know of a good one that will give me, like, I want to know every damned name that comes up as far as because I'm having trouble wrapping my mind around that. I'm not much. I am not good at phylogeny. I'm not good at time. What do you call it?
What do you mean, like the geological column and stuff?
The time periods. That you see charts for. So I have a whole lot of background knowledge that I'm lacking. I got the genetics, but this phylogeny stuff escapes me.
It is pretty lucky that we're debating creationists, and the only phylogenies you really need to know are like mammals. Outside of that, nothing exists.
Yeah, right. Well, humans almost. They don't even want to talk about. They don't want to talk about the bugs.
Well, yeah. Well, definitely not arthropods and stuff. But like, they just want to talk about like humans and apes, and then also dogs and sometimes cows and stuff, and mice. Mice are another one. So as long as you understand Laurasiatheria, you're good.
Yeah, this thing right here. I have a hard copy of this book, and I happen to have a PDF of it. Very interesting. Right around page right here, classification of the vertebrates, and they go through it. Let's see if I can you zoom this thing? Can I zoom? I can zoom.
Most likely, yeah.
Let's see. So anyway, they go through it, and that's what it looks like.
Yeah.
I'm going through it.
I think, honestly, like cladograms and like trees and stuff are much easier to visualize. Yeah. I think these sort of like subsets and stuff, it makes it harder to, like, when you get a subset that's like below 4 other subsets or something,
right?
It gets kind of confusing. You're like, oh shit, wait, is this clade like a dependent of, this clade above it, or like this clade that's like way above it? I don't know. I think it's much easier to see stuff as a tree.
List of boxes. So they've got, let's see, we're looking at some
very early.
Yeah, it's got some pretty good illustrations in here.
Mm-hmm.
Durstone relationships, and then here we go. So what do we have here? Quinozoa.
That just looks like all of the major phyla.
Yeah. So that's all. So that's where we start.
Although I think Metazoa, like oh god, I forgot the Porifera is the most basal, not whatever they had. It was Choanozoa, something like that.
This one. Do you want to go back to that?
Oh, I mean, it's fine. I don't really care.
Oh, Quanozoa. Yeah, so Quanozoa.
Yeah, I think it should be Peripheria, but I'm not totally sure. Oh, well.
Yeah, so this is a crown group. This, would you call this a crown group? Because it shows all the existent.
So crown groups are in opposed to stem groups, right? So you can have a crown tetrapod that's extinct, right? Like dinosaurs are still crown tetrapods.
Okay.
A stem tetrapod would be like Tiktaalik. Or no, that wouldn't even be a stem well, yeah, it'd technically be a stem tetrapod. Or like so basically a clade would be all living descendants of a specific ancestor. So humans and orangutans form a clade of Hominidae, right? That's our family. And then a stem lineage would be a lineage that's more closely related to us and orangutans than it is to gibbons, but it's still more basal than our split with orangutans. Does that make sense?
Nope.
Oh, well, so like Arthropoda would be roughly the last common ancestor of like an ant and a millipede, right?
Okay.
So like that represents Pancrustacea and Myriapoda. And so something like Anomalocaris would be right outside of that node because Anomalocaris is probably more basal to arthropods than like it Anomalocaris would have been more distantly related to a millipede than a millipede is to an ant, and vice versa. But Anomalocaris is still more closely related to arthropods than it would be to like Oncophyrans, which is like velvet worms, or tardigrades, which is another panarthropod. So that makes a stem lineage of Arthropoda.
Who the hell let this guy in here? God, moderation, people. I think he's actually a mod here on this channel. He actually is.
He is.
He is a mod. We trust Standing. My son, Stanley.
Yeah, I definitely do trust him to not post porn in your comments.
Yeah, I don't think he'll do that. So, God knows who's gonna come with him. Oh, Andrew's here. How about that? Hi, Andrew. We thought this would be something that you could contribute to and be interested in.
Okay, sure.
Okay. You woke him up from his slumber.
Yep.
Oh,
so here's
it was just because I was working on some classwork.
Okay.
Well, fair enough. That's what I was doing as well. So
up here we have Craniata, including Vertebrata.
Mm-hmm.
How do you say that?
Vertebrata, yeah.
Yeah, Vertebrate.
And it's Craniata, but I mean, no one really cares as long as you pronounce the syllables.
Okay, so Eurocordata.
Yeah. So Eurocordate. There's usually not an O there, but Euroccordates are like tunicates and sea squirts, if you know what those are.
Yep. So these are. So this back here, this C is basically Cordata.
Well, where? I don't see your mouse.
You can't see my cursor. What is wrong with this thing? So we have Cephalocordata, Eurocordata, and Craniata. Then we have the OI moving backwards to the left, and then we have that big C.
Mm-hmm.
So that big C.
So C, yeah, that would be Chordata. That would be the last ancestor of our phylum.
Then we have Echinodermata and Hemicordata.
Mm-hmm.
Yep. There's more, and they're chordates, right?
So no,
D would
D stands for deuterostome. Echinoderms would be like starfish and sea urchins, whereas hemichordates are penis worms. That's what they're called.
Deuterostomes are
not penis worms.
Deuterostomes are in contrast to protostomes, which include mollusks and arthropods like insects and what else am I forgetting, Walker?
Sorry, you said protostomes. Protostomes are most everything that you can think of.
Mostly everything else.
Yeah. Protostomes would be spiralians, so like annelids, mollusks.
Annelids, there we go.
Yeah.
And nematodes, arthropods, things like that. So
Nephrozoa, kidneys?
Nephrozoa?
Yeah, Nephrozoa. Kidneys?
Where does it say kidneys?
That's what nephro stands for, I believe.
Nephrozoa is, I think that's a fairly large clade. It has like not flatworms, but like different types of like bilateralians. Oh wait, Nephrozoa is just most of bilateralia.
Yeah.
Yeah. Okay.
But why do they call it Nephrozoa? So
Probably has something to do with the appearance of the coelom, if I had to guess.
Nephrozoa, major clade of bilaterians divided into protostomes, deuterostomes, containing almost all the animal phyla. And I'm going to insist on the etymology of this thing.
Like I said, it probably has something to do with like the coelom, which is the tube that goes from your mouth through to your anus.
Right. But nephros means kidney. So
yeah.
I don't know why.
Talking about like the phylogeny of chordates and such here.
We're going to get to, I think, mostly you wanted to get into tetrapod development or fins to limbs.
Yeah, sure.
Yeah,
because I don't know. I just remember you've been mentioning it a lot in your weather reports and stuff. So I had a PowerPoint, so I just said I could talk about it.
It is something I am very interested in. I'm just trying to wrap my head around phylogeny, which I suck at. So I'm trying to find the best way to learn this. And the problem I have with with learning phylogeny is you'll find one chart, and it will not match the second chart. And it's almost like either they've rearranged them, they've changed their mind, or they're giving you a different view, right?
Yeah, it doesn't seem like. When was this book published? Because there's some things in here that are questionable. Oh wait, it doesn't go all the way out to Periphery. That makes sense.
Well, like phylogenies are hypotheses about relationships. So of course they're going to change with new evidence.
Right. Yeah, so it's really hard to learn to wrap your head around it unless you can...
except the major transitions, the major groupings are pretty stable for the most
like this, like the Nephrozoa and deuterostomes and protostomes.
Yeah. We're not gonna say plants are more related to fungi. That's probably not gonna change anytime soon.
Yeah. So the major. Usually, I mean, this is just a general rule. The stuff that's studied the most is the most well understood, right? Yeah. So, we know a lot more about mammal phylogenetics than we do about ctenophores or all these other things that no one cares about, like hemichordates.
So, what year would you guess this book is from?
2004. That's going to be my round guess.
2015.
Really?
Yeah, okay. It's most current edition. By
maybe it's the black and white that tripped me up. I don't know. Things that are black and white just look like.
Hey, Walker, don't judge a book by its cover.
Fair enough.
Well, it's got this fancy color cover way up here. I mean, and they've got colored plates in the back. So
does it mention anything on outgroups by any chance?
In this book?
What do you mean?
Yeah, an outgroup is basically okay, so the group you're studying, and I kind of learned this recently, actually, but the group you're studying is called the in-group, okay? It's the group all the characters you're looking at. An out-group is a related clade that you're not looking at, but that from other evidence or other studies is closely related to the in-group, and that can help determine the root of the tree, for instance, whether... go ahead.
Oh, well, I was just going to say that you have to have 3 species for comparative phylogeny, right? Because if you just have 2 species, and obviously you assume relatedness because all evidence that we have points to relatedness.
Right.
All you have is a triangle, right, where they connect, and you can't really dictate where they connect compared to other things. So so basically what you're saying, the in group is any 2 nodes that you're specifically looking at, whereas the out group would be just another node that's outside of that clade.
And if I may add, the phylogenies tend to be made up of multiple outgroups, or sorry, they tend to include multiple outgroups to decipher which characters are ancestral and which characters are most likely synapomorphies.
Yeah.
And this can what else was I gonna say? Darn, I forgot. Oh well.
Well, yeah. I mean, if you're trying to root it, so it's called rooting a tree whenever you include an outgroup.
Right.
And usually that's done in phylogenetics. So like when you're taking genetic data and putting it into a phylogenetic tree. But you can also root it for like derived synapomorphies. I don't think they would do that for the textbook. And also when you're using a clade as big as like Metazoa, which is a fairly well-established clade, I don't know if you would, but like if you were doing Arthropoda, you would probably include an Oncophyrin, just because they're relatively closely
I was
related.
just asking because recently I was looking into the idea of transitional forms being used as outgroups, perhaps, or like fossils in general.
Yeah, that would be interesting. I mean, you could definitely do that with like Australopithecus afarensis compared to. I mean, that's how you find the derived synapomorphies in human evolution and stuff, right? Like you compare, what did Australopithecus afarensis have that Ardipithecus sediba didn't, or not Ardipithecus sediba, fucking standing, man. He got me on the sediba train. No, Ardipithecus ramidus, you compare those and you're like, okay, well, here's some derived features that they have. Okay, what about Africanus versus afarensis? What about, Africanus versus Homo habilis versus Homo erectus until you get more and more human-like, essentially.
Yeah.
Standing
side route.
Standing
you're good.
Of God in the chat. So Standing is doing God's work. He's trying to distract everybody by giving them good things to watch on Netflix.
Wait, afarensis is a mixed species now? I thought it was Sediba and Homo habilis.
They changed.
Yeah. I don't know. Like I said, I feel like the fossil record of humans is so well understood at this point that you sort of have to artificially create holes in it so that you can say, okay, well, here's the line between kinds.
Yeah, not by this guy. I mean, this is...
What the heck is a mixed species? I have no idea what that is.
So his idea of mixed species is when, you find bones and, some of these bones are ape bones and some of these bones are human bones, but they assume they're from the same specimen.
That doesn't happen very often.
So he's got this Lee Berger article that he keeps talking about that at some point we'll have to look up, but we don't want to get distracted by Stanley right now.
Yeah.
Yeah.
Yeah, I could screen share my PowerPoint if you'd like.
See, he's trying to distract people by giving them shit to watch on TV, so they can. Stanley, shame on you, Stanley. Okay, you want to do your PowerPoint?
Yeah, just give me 1 second to pull it up.
Okay, we'll make some more sense out of things.
Sorry, I just want to mention outgroups because I find the phylogenetic stuff is pretty fascinating when you get into it.
Yeah, there's a few things it's difficult to wrap your head around at the beginning. But this page 37 of this vertebrate group, they get into outgroups and what exactly the meaning of them are and paraphyletic groups and all of that. And then they also do a nice little diagram on the bottom, which we've seen 100 times. And I'm still wrapping my head around phylogeny. And once I get something, it locks in. I can feel it lock in. But this has not locked in, and I have trouble with it.
I think the best way is to just look at a bunch of bunch of trees and stuff.
Wait, Speed. Is your mind full of Legos?
Yes. Yeah, they lock together. Like the interesting thing, if any of you ever go on to teach people, the most important thing. That's okay, Stanley. I forgive you. The most important thing is to create an overall structure and put that in the person's head. So if you want to understand phylogeny, you're going to want to see the broad strokes and you're going to want to memorize that structure. If you're trying to understand the anatomy of the brain, then you've got to understand those lobes and you've got to be able to put them together and you memorize that structure. From then on, everything that you learn, all the details, they have a place to lock in. And when they lock in, you will end up with really good, solid learning. If you do not establish this overall framework for your students, like if you just start telling them how to differentiate polynomials without having some overall structure, they're never going to learn.
Right.
It's going to fall apart. Every time they learn something new, it's going to tear down the old structure because they have no place to hang it. So anyway, that's really important. So what I would like to develop, or what I have to develop for phylogeny, and I would like you two gentlemen who know more than me to help me with, is to lock in that structure at some point. So anyway, but moving on, we have a presentation by Just a Walking Fish.
yeah. No. Fuck, how do I make it not pop up the
I think, is this PowerPoint?
Google Slides?
Oh, Christ.
I mean, I can just, if I have it open on this slide. Okay, yeah. I mean,
anything that starts with Google always ends in tragedy.
It's true. I don't know. I like the open sourceness of like Google Docs, or not open source, but like how editable it is and like how shareable all that stuff is. It's a lot easier for like presentations. Yeah. So the most basal clade of what people generally consider fish. Some people consider like sharks fish, but most people wouldn't consider rays fish. So I'm excluding cartilaginous fish. Most people would consider like osteichthyes fish. So that's like bony fish. And there's two major clades in osteichthyes. It would be Actinopterygii, which is, your ray fin fish, and pretty much everything you think of when you hear the word fish. So goldfish, trout, tuna, flounder, cod, all of them. Whereas Sarcopterygii actually only has 8 species that you would consider fish, and the other 30,000 are not very fish-like. But cladistically, obviously, I would still say they're fish. Hence my name. An interesting fact about this clade would be that the lung actually probably developed basal to this split, because there's very primitive Actinopterygian fish, such as beachirs, that actually have lungs. And then other lineages of Actinopterygian fish, like teleost fish and stuff, it actually was adapted or exapted into a swim bladder. So the lung predates the swim bladder. And that's a very common misconception that people have. People usually think that the swim bladder predates the lung. That's just not true. Yeah. So within Sarcopterygii, they're generally considered the lobed fin fish. This is Guiu, one of the oldest lobe fin fish that we know of, and honestly one of the oldest bony fish that we know of. They first come from the late Silurian period, about 420 million years ago, and they were pretty successful from essentially the Devonian. And they tapered off a bit during Carboniferous. That was mostly the time for sharks, same with Permian. And then they became really, Sarcopterygian fish became very prominent again for the Triassic and Jurassic. There's some debate about like what the actual earliest Sarcopterygian fish is, because, when you're at the very base of a clade, a lot of the fish, a lot of the organisms seem very similar. So like the earliest Actinopterygian fish are very similar to the earliest Sarcopterygian fish. But Guiu is the oldest one that we're fairly confident about. Oh, sorry.
I'm just looking it up in my book at the same time. It's on page 72 of that book I showed you.
Oh, wow.
Guiu. So
yeah. Oh, is it actually? That's funny.
Oh, it is, yes.
So the most basal living clade of Sarcopterygii, and one of the most basal clades within Sarcopterygii in general would be Actinistia, better known as coelacanths. And coelacanths they first appeared in the late Devonian as well, but they were most successful from that time period through to Triassic, Jurassic. And they were thought to have gone extinct in the KPG extinction, but they're now considered a Lazarus taxa because we fairly recently discovered one in, I think, 1938. So what was I going to say? Oh yeah, so a lot of creationists always consider coelacanths like the evolution defying fish or something like that, because I don't know, they're a relatively unsuccessful taxa today. Like there's very few of them, and they supposedly don't seem to have changed very much morphologically through geologic time. But I included this phylogeny on the right, I think, to show that point.
Can I very quickly share my screen and then we'll switch back to you?
Yeah, go for it.
Okay. Let me see if that pops you out.
I popped myself out.
Okay. So this is a nice little diagram of what you're looking at, the Utilistome, Actinopterygii. Rigii, you always say E.
Yeah, Sarcopterygii. It's
Rigii.
Actinopterygii.
Yeah. Okay. And then the Crossopterygii, and then those are the coelacanths, and then Rhipidistia. Okay.
Yeah, Rhipidistia is the next one.
Yeah. Hypnomorpha, and then tetrapods.
Mm-hmm.
So this gives you a nice little diagram. That's what I mean by having something to hang your information on. So too bad we can't put more than one thing on our screen at the same time. So you go ahead. I just wanted to present that.
Why are you showing me this? Everyone knows a coelacanth can't give birth to a non-coelacanth.
Yeah.
Oh, so I will go back to screen sharing. Sorry, I dropped mine so that you could put yours up.
Dr. Dan's here. Hi, Dr. Dan.
Hello, Dr. Dan.
Hello, Dr. Dan.
Throw the invite out if you want, but yeah, and this was something else I found whenever I was like first researching this. So Latimeria is the living genus, the extant genus of coelacanths, and I found it extremely interesting whenever I was looking at them, because these two species, which look very similar, actually have about a 40 million year divergence. And there's a population of the Indonesian coelacanth that has about a 12 million year divergence from the other population of Indonesian coelacanths, right? So like in comparison, these two species of coelacanths are as closely related as we are to like New World monkeys. And within the population of Indonesian coelacanths, they're as closely related as we are to like gorillas, right? And they're still considered the same species. So I think this is a perfect example of one, evolutionary stasis, obviously, and two, how the species concept is completely bunk. It's just not. It's not as accurate as like representing stuff with lineages. And three, I think it's really interesting because, you know, supposedly these examples of evolution-defying organisms are so completely divergent, right? Like this isn't something you shouldn't expect any species to have a 12 million year divergence within two populations. You definitely wouldn't expect a created kind to have as much genetic divergence as like Old World and New World monkeys, you know, because we are definitely not the same created kind as like a squirrel monkey or a spider monkey. So I don't know. I just think that's something kind of interesting.
Yeah.
That these are so diverse. Well, not diverse. They're very small populations. Divergent. It's a better term. So yeah, here's Rhipidistia. You were talking about it earlier. So Rhipidistia is a sister to Actinistia, which, you know, obviously Actinistia contains coelacanths. So Rhipidistia is all things that aren't coelacanths that are still Sarcopterygian. So it would be like us and lungfish, pretty much. They have some pretty interesting synapomorphies, actually. So the most obvious one would be the lungs, right? Other than the Queensland lungfish, which is the most basal lungfish, the other 5 species of lungfish are all obligate air breathers, so they would actually drown if you held them underwater, which is pretty funny. And they also have fused upper jaws, like us, right? If you've ever seen like a fish eating, you'll notice that they extend their upper jaw outward. You know, lungfish can't do this because it's fused to their skull. Right, and obviously we can't do that. Our upper jaw is fused to our skull, so that's a cool thing. If you notice the South American and African lungfish, the most derived lungfish species, they have a lot like wispier arms. But if you actually look in the fossil record, the oldest lungfish that we know of had much more fleshy limbs, more similar to that of like coelacanths. And this is pretty much the last, you know. Well, this is obviously a fish versus, well, I don't really know about that, clade. You know what I mean? Like, after this point, it starts becoming a lot more gray as to whether or not a species is a fish or a tetrapod, because we're in Tetrapodomorpha. Which go ahead.
Where does a bowfin fit in?
Bowfin fish aren't Sarcopterygian. Bowfin fish
They aren't.
are definitely Actinopterygian. Let me see what specific clade they're in.
Because those things crawl.
Oh, okay. So I just looked it up. Bowfin fish are relatives. They're in Holostae. I think there might be labyrinth fish.
They are Actinopterygians.
Yeah. So there's actually a different clade of fish in Actinopterygii that has evolved something called a labyrinth organ, which is like a specific channel within their mouth that they can diffuse oxygen into their bloodstream. So it's like a lung inside of their mouth, essentially. And that's also snakeheads are also in that clade, and also like gars. I don't know if that's true. They might be a different clade, but I know like what the archer fish. I'm pretty sure they're labyrinth fish as well. I don't know.
We had a question from Nestleg. I'm sorry, guys. I probably missed a lot of chat. Just keep trying to get my attention. I don't have an attention bone. I'm a non-whatever.
I've seen somewhere that early tetrapods have decoupled respiration, took out oxygen with lungs and respired CO2 with gills. I've not heard on that. I can look it up.
I did hear something on this. The tetrapods that decouple respiration.
I don't know. That might make sense because they probably didn't have like a diaphragm yet. But yeah, I have no idea. Yeah. But yeah, so tetrapodomorphs are basically, so this would be an example of a stem lineage, which you were talking about earlier, right? So no tetrapodomorphs are alive except for true tetrapods. And true tetrapods would be the last living ancestor of us and a frog and all of its descendants. So these are all technically stem lineages to tetrapoda. Does that make more sense in context, Speed?
I was typing. I'm sorry, I wasn't paying attention. Put me in the corner. What did you say?
Oh, well, I was just asking if so Tetrapodomorpha, like these two species that I have on the screen, like Megalichthys and Rhizodus, would be an example of a stem lineage, right? Because these are lineages that aren't ancestral to any living population, but they also don't share common ancestry with the current members of the lineage, right? So like they aren't true tetrapods, but they're more closely related to tetrapods than they are to lungfish. So that makes them stem species within Tetrapoda.
So
Tetrapodomorpha.
Morph fishes, and the other branch is Tetrapoda.
Kind of. It's not necessarily two branches, because once you get into stem lineages, the actual branches look a lot hairier, if that makes sense. Like they just have a lot more tiny branches that end really soon after they appear. And that's just a general concept of, most species that ever live are going to die out. So the chance of any one specific lineage surviving is pretty low. And the chance of that one specific lineage being ancestral to all of those current descendants is a lot lower. Does that make sense?
Yeah.
Yeah. So Rhizodus and Megalichthys, Rhizodus was in a family of giant freshwater fish called Rhizodidae, and they all went extinct. They don't have any modern descendants.
Okay.
So that makes them, just a small blip on the tree of life compared to Tetrapoda, which is now a pretty prolific clade.
This is really good advice.
Which one? Just ignore standing, focus on the conversation.
Standing is trying to pull us off in aphorensis and all this stuff. So anyway, but we do want to have you on soon, Stanley, to educate us, because we are so clueless. Apparently. Yeah.
Oh, and there's another, so like a derived synapomorphy that unites this clade. You can sort of see it would actually be nostrils, right? If you look at Megalichthys, and you can sort of see the one in Rhizodus, right? So Tetrapodomorpha is where the first nostrils appeared. Although the nostrils probably weren't involved in breathing. I don't think they directly went to the lungs, but they would have had an olfactory role, so that they would have been able to sniff out stuff, which makes sense because the habitat for these dudes is likely murky waters, like lakes and stuff. And Rhizodus was actually the biggest freshwater fish that we know of.
But they had to be able to pull in water right through those nostrils. So that's kind
Well,
of
they probably breathe through their mouths, if I had to guess, or their fins.
Yeah, but how do they get in? Okay, when they developed these starting nostrils, they had to be able to pull water in through those nostrils, and that was probably the beginning
of like
of
respiration.
forming a connection.
Well, I don't know. I don't know about that. I mean, they definitely need to have water going through it, but it could just be like a passive thing where it's
from moving
particles. Yeah.
But you see what I'm saying here? That at some point, nostrils became connected with the airway.
Mm-hmm. Yeah.
In tetrapods. So what is the base of how that happened? If it was olfactory, then there was a reason to pull air or water in, and that would have connected up with whatever it is that pulls water into some other orifice, and those two orifices connected at some point. You see what I mean? Developmentally. Look at them how it developed.
That makes sense. I don't know of where that would happen in the fossil record or anything.
Yeah, that's
just because I mean, like, I like looking up this stuff, but I am by no means an expert. You know, if you talk to like an actual fish paleontologist like Neil Shubin or something, he'd probably have a much better answer.
Stanley, you can't block Dr. Dan. Dr. Dan is the one person that actually has critiques for you. I mean, come on, Stanley, my son. I raised you, but no, I didn't raise you. That's right. I went out to get ice cream for his mom, and I met Gavin, and I ran off, and I didn't raise Stanley. That's how I abandoned him. I don't know if you got the backstory or
not.
The father or whatever, the parental connection there.
Yeah, the way I abandoned my son Stanley. I went out to get ice cream for his mom, and I met Gavin Herleman. He was so hot. I ran off with Gavin. Because he didn't have a shirt on.
You went all the way to New Zealand.
That's my story.
That spicy hunk of meat.
Well, he paid me some money to go to New Zealand, but we won't get into that.
Yeah.
Okay.
Yeah, so Rhizodus was a massive fish. It would have dwarfed a great white by like over a meter. And it was a lake fish as well as a freshwater fish. So crazy. I don't know. It's a true river monster. So Eotetrapodiforms, it's just another clade of stem tetrapods, essentially. But these are where you start to get into the fishapod territory, right? So like any of those cladograms that you've seen with like the progression, they always start with Eotetrapodiforms, usually Eusthenopteron, sometimes Panerichthes. They still look very fish-like, right? Especially the top two.
Yeah.
But they have some very unfish-like characteristics. So Tenorau, which is one of the oldest, or not necessarily oldest, but like most basal Eotetrapodiform, it had actually a shoulder socket, and it had a tibia and a fibia and a fibula, which is pretty interesting. And then Eusthenopteron, which is a little bit more derived. It also had a humerus, an ulna, and a radius. So at this point, you already have the major arm bones, and now you just need to structure them properly. And Panerichthes is one of the most derived Eotetrapodiforms, and it's pretty obvious. You know, you start to see the what is it, ventral flattening, dorsal flattening? I don't know. I need to take an anatomy class. But you know, you see it start to flatten out compared to the streamlined fish body plan. And it didn't have an open intracranial joint. Okay, so the intracranial, fish have a joint on the back of their head. Let me see if I can. If you look at the left cladogram right here, you can see that there's a joint that separates the skull from the neck. And we know that Panerichthes actually had this joint starting to fuse. And obviously we also have it fused, right? So that's one of the derived characteristics that Panerichthes had. But it still had the intracranial joint on the inside of the skull case. And then Panerichthes also had the beginnings of limb, or they contained bones that were like the analogs to digits, right? So this is where we start to see fingers appear. And then Panerichthes also had a very primitive pelvic girdle, which is interesting. So
now they recently found the precursors of fingers in paddlefish, which are actinopterygi.
Yeah.
So they pushed it back a little bit, and they were a little surprised by that.
Yeah, so I think that's where you were bringing this up, and I thought that was really interesting. And I haven't read that paper that you've been talking about.
And they had
is it that they found the genes for it, or is it that they found the actual bones? Because I don't think they found the bones.
They found actual precursor structures and Hox genes that promote them, and they found them in modern paddlefish. So they pushed back. So they used to think it's called the autopod, your hands and your wrist and all that.
Mm-hmm.
And they used to think that came after the split between Sarcopterygians and Actinopterygians. And now they pushed it back further to that common lineage because they found basically basal mechanisms to produce those fingers.
Well, but the earlier fish don't necessarily have those structures like present, right?
No.
Like they just have the genetic pathways that are like precursors to it.
Right. And so they're not sure if they had, if some of the basal lineage had some of those structures or not, because they'd have to find fossil evidence for it, but they definitely had the right Hox setup in order to create them. Mm-hmm. So, and because they popped up in both branches. So
yeah,
and that was Nephilim Freeze slam dunk proof against evolution.
Well, that's a very interesting example of like genetic exaptation, right? So that's the whole
well, that's a really good example of evolution. I don't know why you thought it was proof against it.
I mean, everything in evolution has a precursor, right? You're not going to, as much as creationists would love to like point out like, oh, well, you've never seen a gene just magically appear out of nowhere. Of course, that hasn't happened, right? You don't have proteins just forming out of nothing. Everything comes from precursor pathways. And that pushes it back to abiogenesis, which is why I don't like arguing against abiogenesis, because I don't know that much about it. But like any pathway, so a perfect example of this would be the citrate development, right? Then the long-term evolutionary experiment. The E. coli strain that eventually evolved citrate metabolism had a precursor duplication event around generation 18,000, and they developed the citrate metabolism around 30,000. So it took them 12,000 generations to get whatever the right mutation is for the aerobic fermentation of citrate. Or not fermentation. Oh, wait, it is fermentation, yeah. So the aerobic fermentation of citrate. And they found that any of the precursor populations before the generation 18,000 weren't able to develop citrate. But the ones that came after it, most of them did eventually develop citrate metabolism.
Yeah.
So that's kind of the topic of what my second video was on. It's an idea known as like evolutionary determinism and how much your evolutionary past will influence your evolutionary future.
Yep.
So those are pretty cool concepts, but I digress.
Yeah, and that's probably what happened in that basal lineage that may have had those bones form for a shoulder clavicle, and it may have been for some different articulation of the fins, so that the fins could move, and they may have been bottom dwellers.
Yeah, so that's the current thought. Most of these, like Pandrictes and Tiktaalik, were probably still fully aquatic, but they probably lived on the bottom of the like riverbeds and stuff.
Yeah, and they could crawl along on the bottom, which rightly led to crawling around on the shore. So
yeah, well, yeah. So I mean, we know that Pandrictes, it did have a pelvic girdle, but its pelvic girdle wasn't weight bearing yet.
We have accomplished something today. We have brought these two back together.
Oh my God!
We have seen the
doing the Lord's work.
The rejoining of a ShamWow salesman with a doctorate of biology or doctorate.
Yay!
Yeah,
and virology.
Sorry,
Stanley. Genetics.
Oh, I love to give Stanley shit when he's over here. He takes it well.
Yeah, and anyway, so should I continue on to Stegocephalia? Interesting stuff.
Who would like that? Who would love that?
And by the way, guys, this is why I don't do. I usually script everything I say.
Okay.
Because I know I'm boring as fuck.
No, you're not.
Yeah, so this is Stegocephalia, and this is what generally people refer to as fishopods, and these are the ones that are extremely characteristic because most of the major transition from fish-looking thing to land vertebrate-looking thing happened in Stegocephalia. And once again, all of these are stem lineages, right? So, like, Stegocephalia isn't necessarily a true... Well, Stegocephalia is a true clade, but, like, these don't all nest together, right? We're much more closely related to Ichthyostega than we are to Tiktaalik.
Right.
Yeah, I just keep hammering the whole stem lineage versus crown lineage thing home because Speed was asking about it.
And you also gotta...
Oh, go ahead. No.
Oh, I was saying you also gotta point out that just because it's a stem lineage doesn't necessarily mean that it's a ancestral lineage. You know what I mean?
Oh, well, yeah, that's especially true for Tiktaalik, which is like one of the biggest misunderstandings for it. Because every creationist and their mother will say like, oh, well, Tiktaalik can't have been ancestral to us because we have, fin impressions that are from earlier. And it's like, okay, no one has ever said that Tiktaalik is a direct ancestor of vertebrates, right? It's a very good example of a transitional form, right? It has a very basal body plan with structures that are intermediate between, eotetrapodiform and true tetrapods.
And if I may add something onto that, cladistics in the most general way doesn't rely on time to group organisms by their similarities. Like it's just a way of grouping organisms. It doesn't necessarily rely on the time axis or the order of the fossils in the fossil record, for instance.
Yeah, absolutely.
Yeah, it's basically just comparing character states across a matrix, which I found was pretty interesting.
The Earth absolutely could be 6,000 years old, but it would have to mean that humans evolved like 2 days ago. You know what I mean? Like, right. Evolution has no implications for time other than you have to have time for everything in the fossil record to occur.
Stepping away, guys. Stepping away for a minute.
Oh, okay, yep. But yeah, all you need is like, the dating and stuff that comes from geology and all those other dudes. Biology and paleontology mostly just talk about like the stuff that happened and where it was happening and when it was happening as the dates were taken from someone else. You know what I mean?
Yeah, I think it's fair to say that evolution entails relationships, not necessarily direct ancestry. Like that's kind of the mentality in cladistics, instead of something like this is probably a bad example, but like Aristotle's great chain of being, for instance, or the Scala Naturae. I'm probably mispronouncing that, but yeah. I don't think that Aristotle actually believed in evolution, just a side note, but kind of that linear progression, I think, is what most creationists have in mind when they think of phylogenies, you know what I mean?
Yeah, the whole march of progress thing.
Yeah.
There's a great quote by Stephen Jay Gould. It's like life is a copiously branching bush. And I think that is the best analogy for life, right? You just have tons of little twigs going off in all directions, and it's like you can't really predict which way it'll grow, but it'll grow some way. And most of these tiny little twigs aren't going to go into these huge networks, but they might. And I don't know.
Maybe a good way to think about it isn't necessarily the upper half of a tree, but rather the lower half,
like the root system.
Yeah, like the roots are kind of spreading out and conquering new niches and things like that, and maybe that's kind of an equally good analogy for that as well as the tree analogy.
Yeah, but yeah, and I guess back to what you were saying earlier, cladistics is never going to try to assign direct ancestry unless we have rock solid evidence of direct ancestry.
Like I think there are specific fossils like foraminiferans. Am I pronouncing that correctly? That like you find them in stratigraphic layers right on top of each other, and basically that.
Yeah. Oh, those are like the small shelly fauna.
Yeah.
Yeah. So that's actually, and like the diatoms and stuff like that, and like there are certain fossil species that we don't have any genetics for, but we can fairly confidently say that we like a species evolved from. So we can say pretty confidently that the woolly mammoth and the Columbian mammoth probably evolved from another species called the steppe mammoth, right? Or like humans our ancestry eventually goes back to Australopithecus africanus, and that's probably our last common ancestor with paranthropines, right? But it was like that specific species. But that's a very rare situation, and I think that's usually because we have such good gradients and we have so much. Elephants and humans are probably some of the most well understood fossil records, especially within the last 2 or 3 million years.
Well, I would think that gradients would actually kind of make it even more branchy. I mean, right?
Yeah, it does because most populations go extinct. Well, that's the thing. We would prescribe our ancestry to like Homo erectus is a great example of that. We know that we haven't found a population of Homo erectus that is a direct ancestor, right? But we know that if we found the direct ancestral population, they would have been classified as Homo erectus. Right. Because we have Homo erectus that are more basal and then Homo erectines that are more derived. And they're still considered Homo erectus because it's an evolutionary grade, not a true clade. Right. But, those are the exception that proves the rule. I can only name like 3 or 4 species like that. And everything else, if you see it in a cladogram, it's not going to be direct ancestry. Right. Yeah. Okay. Yeah, I'll get back to Stegophilia. I'm almost done, so I'll try to wrap this up pretty quick. Stegophilia, which means, these are the fishopods, essentially. 2 of the biggest synapomorphies would be the intracranial joint is entirely gone, so their skulls are pretty much like ours. Their skulls are fusiform, right? So their upper jaw is attached to their skull, and their skull is one big piece. And then also the pelvic girdle, or not the pelvic girdle, the pectoral girdle has separated from the skull. So that means that these fish also have like not just shoulder sockets, but they had true shoulders and they also had necks. So we're looking at fish with almost the same body plan as, humans and other land vertebrates, tetrapods. Tiktaalik is actually one of the most primitive stegocephalians. So, and it chronologically existed later than most of the other derived forms, so it wasn't a direct ancestor. But it has all of the morphological features that I just talked about because it's the most primitive stegocephalian. It also had like primitive wrists and elbows, as well as a reduction of fin rays. Fin rays actually aren't homologous with fingers, so that's another interesting thing. The fingers are homologous with bony structures that are inside the lobe. Had a robust rib cage and longer limb proportions. So it was probably spending a lot of time on the bottom of whatever habitat it lived in, but it probably didn't go up on land unless it was like had to go from a pond to a different pond or something like that. But it wouldn't have been a very efficient walker. It probably couldn't have done anything on land except flop around like a snakehead,
You wouldn't have been an efficient walking fish. Ha ha ha.
Yeah, not at all. He should just grow legs like mine.
So what do you think, Dr. Dan and Stanley, mud wrestling?
That'd be fun.
Yeah.
I would watch that.
I'd watch that. On my channel. I want it on my channel.
I want to see Raw Matt wrestling someone as well. Or not like wrestling, but like cage fighting. Yeah.
I saw a picture of Stanley's arms. He's pretty well exercised.
Well, he's pretty open about it. He wrestled in high school. I think he was like a champion, like a state champion wrestler or something.
Yeah. Yep.
So I would not want to go against him in the ring. Just the ring of debate. No. So the next fish pod. So Acanthostega is intermediate between Tiktaalik and Ichthyostega. And it roughly follows the same general trends, right? So the limbs are longer than they were in Tiktaalik. The fin rays are gone. They have a reduction of the number of bony protrusions. So they actually have 8 of what we would call fingers. And its pelvic girdle was also likely weight-bearing. So it could go up on land, not well, and it probably couldn't move around on land, but it could use it as an escape option, right? Like it could flop up on land if it was getting chased by a predator and then go back into the water. And then the last fishapod before we get to like true tetrapods would be Ichthyostega. which is almost a true tetrapod. It has some basic differences that I won't go into too much. It had 7 fingers instead of the tetrapod's 5 fingers. Like all of us have 5 fingers. And even the tetrapods that don't have 5, it's the basal condition for those tetrapods. And something that's interesting actually is that they wouldn't have been able to walk like a salamander where they do that little like squirmy thing. Instead, they probably would have walked, or they would have flopped around on land like a seal, which I find really funny because they had really strong pectoral muscles, but their pectoral muscles couldn't necessarily bear weight. They could just flap really hard. So these guys would probably get up on land and then bounce around like a little seal pup, which I think is just very cute. Yeah, and at that point, I mean, you know, we have things that are walking around on land, albeit not very well. They have, you know, derived arms and legs. If we saw an ichthyostega today, most people would probably say like, oh, it's a tetrapod, you know, it's just some sort of amphibian. Yeah, and so here I have some cladograms of the transitions. So as I already showed on the left, that's the transition of like the skull joints and stuff. So I think that's mostly the intracranial joint. And the one on the right would be the fin, or like the fins to hands and stuff. And so acanthostega is the one on the far right, whereas a coelacanth is the one on the far left. And, you know, you can see like how the different bones are developing from, you know, I don't know the actual bones, but like big bone, like 1 bone, 2 bone, lots of bones, that sort of thing. That's, I think, Neil Shubin's thing. You can definitely see how it's progressing through time into a more tetrapod-like limb. And so here we have like some of the most basal tetrapods, which, you know, are still like even arguably tetrapods because cladistics is a hard thing. You know, you can't really say like, okay, here's the cutoff, because there's always going to be a species that's slightly more basal and slightly more derived. Yeah, so something like Tulerpaton, it's arguable if it even was a tetrapod because it had 6 digits instead of 5. And it was definitely an obligate air breather, though. So, like, it was definitely, it couldn't breathe water, right? Even though it was fully aquatic, probably. And then you have, like, anthracosaurs, which would have been, like, the earliest land animals, right? So they're probably on the reptiliomorph side of the split, actually, which means they would be slightly more closely related to us than a frog, or like a salamander, or Cilian, whatever those little wormy things are.
The Cilians?
Yeah. So this is specifically Samoria. It's a species of anthracosaur. You know, it would have probably lived its entire adult life on land, but it would have still had to go back to the water to breathe because it's definitely not an amniote yet. And they had 5, you know, digits like all of us, right? So we do know that the pentadactyl hands were fixed in the population at this point. Yeah, that's pretty much the end of my presentation. I have the 4 sources. I mean, these aren't the sources I used to necessarily compile the slideshow, but these are some cool ones, including the 3-dimensional limb joint mobility in early tetrapod ichthyostega. That's the one where they have them like flopping around like a seal. It's a cool one. And then the rest of them are all about coelacanths, really.
108, 109, Samoria. Interesting.
And I feel like I almost copied your vertebrate paleontology book.
Yeah.
We convergently evolved on the same strategy.
Yep. Are you done?
Yeah, that's my whole PowerPoint. That PowerPoint was just something I dug out of my Google Slides because I don't know. I kind of wanted to talk about like actual evolution instead of just the evolution creation debate.
Yep. The basically when a limb bud appears, this is what it looks like, and it's got this apical ectodermal ridge, and these basically like from well, let's see. Can you see my cursor on this thing? Why can't you see my cursor? I can't imagine why. God, that's too bad. Going from, you know, from right to left where the body is, there's basically these fields that develop, these axes that develop, and like on one side, on that apical ectodermal ridge, it's giving off, basically it's called paracrine enzymes. They sort of float out, they leave the cell and they enter the matrix around the cell. And then on the left-hand side, there's a different enzyme, and there's a field that develops between them. And they sort of start to create these. I got a bunch of pictures here that I don't remember exactly what they are. This is from Schubin. Okay, basically the limb, but let's see, where are the Hox genes? I'm trying to find my Hox. These are fields, basically, these colored things, and they determine how each part of this is going to develop. Mm-hmm. And it's really interesting. Then when you go down to one finger, these Hox genes again form another field from one side of the finger to the other, from the tip of the finger to the base, and they do some more delineation. And the thing about these is very little modification in genetics. Like just one transcription factor having a couple of amino acids change can drastically change the expression of the genes along an axis. Like you got to think in terms of a three-dimensional axis, and there's so on each end of it there's like a little signal molecule that's sending signals across, and in the middle you'll have 40% of this and 60% of that, and then 50% of this and 50% of that, and that sort of kicks off the whole process, and that's how Hox genes do this stuff. And there's I got, let's see, various pictures of limbs. Trying to find.
This is where he keeps his bone collection.
I keep my bones here. Basically, there were some real stunning ones that I don't quite have yet. Maybe, let's see, what is this? A pubis. Trying to figure out what this is. And I'm not ready to have this talk yet, but I'm trying to get ready, basically, because you can see back and say, like early fish, there are. At the base of the tetrapods, there are little cartilages, like little clusters. They're almost like little round circles, and they expand into each of these bones that you see: the tibia, the fibula, the tarsals, metatarsals. And it's really interesting how you can see the progression when you look at a number of different species. You can almost see how it progresses. And for extant species, we can study how that develops. And we could determine which genes were necessary, and from that you derive the story of evolution. And it isn't something that we cannot figure out. It's just something that takes a great deal of time to figure out. So
right, yeah,
I'm working on that sort of. What else do I have here? I got some other. I have some amazing, scary looking pictures of fishies.
Getting ready for Halloween.
Yeah, get ready. Oh, Spooky Bed Hair hasn't popped up yet, has he? God, that guy scares me.
Spooky Bed Hair? Oh, yeah. Who is he? He's like a fairly new, like a recent arrival, right? I don't know.
Maybe 6 months, a year? I'm not sure.
Well, I don't think I've seen him before. Is he creationist or what?
I don't think so, no.
Huh.
Pretty sure he's...
You should be a fishapod for Halloween.
Fishapod. Actually, that is my plan. I have my fish mask over in the my drawer over there.
Well, I thought of dressing up as a basically what is it, a 70-year-old man with a cough.
Mm-hmm.
That would be the scariest thing you could ever find for Halloween.
Yeah,
a low raspy cough, you know. And pale skin.
A dry cough.
It's like COVID zombie. Oh, the dress up was like old COVID zombie. I think that'd be great.
Yeah. Oh yeah, that's another reason I've been incredibly busy. Somehow all of my family just tested positive for COVID like in the past week.
What? Oh, no shit.
Wow. Yeah, it's fucking crazy. I mean, yeah, they're all doing fine. My uncle and my aunt are both basically asymptomatic. My dad, I think his fever is over 100, but he's on the like downhill slope, so I think we're good over there.
Oh,
yeah.
We'll hope they get better.
Well, the Rage Nakasuchi, he just got sick again.
He got sick again?
Yeah. It hits you in two waves. Like you'll start to recover, and then you go back into it. And so I'm not sure how he is.
Mm-hmm.
We just heard about that this morning, so it can be pretty bad shit. What was I going to say? God damn, it was something. Oh, when I go to a store and they're like if I have to walk into a gas station, which I do occasionally, and there's somebody else in there without a face mask, you know what I like to do? Start coughing.
Mm-hmm.
You'd be surprised how fucking fast they back off.
If anyone tries to mug you, you can just cough on them.
Just start coughing on them, yeah. But I mean, these assholes that go in without a mask. I won't go in the store if I see one, but every now and then one will slip in. And you just give them a couple coughs and boy, they back off about 10 feet real fast. It's real nice. And given my allergies, I can always bring a cough to bear. Plus, I look old and scary, so it's really cool. I really enjoy it. That's what I'm going to do for Halloween. Go around scare little kids with my cough.
Mm. Yeah, you cough in their candy right before handing it to them.
There it is. I can cough. Cool. Anyway, so I would like to put together a more detailed presentation of how fins and limbs develop, and I have a tremendous amount of. Here's some interesting stuff. What the hell is this? Why can I not see that? Oh, because it's. What is this thing? I don't even know what that is.
Is that Darwinian?
I got some primates. I got some primate pictures laying around. They're just much bigger than the screen is allowing for. I guess this was some other stuff that I just had collected for some reason. Standing's going to get excited if he sees this. I better hide it. Gotta hide that. Anyway, so yeah,
was that like macrobats versus primates or what?
Which was that? This one?
What was that last slide that you had?
Is this one of the ones from Raw Mat? I'm not sure. This is diverse later Pleistocene homocrania in anterior view. So I'm not sure where I got this from. Probably one of the papers that I got from something Stanley posted. So I don't know. Because I like to grab the pictures. Ooh, that's a cool looking dude. Well, he got an insect. Nice prize. Very good. Let's see. What else do I have?
What is this from? Is this from the Vertebrate Paleontology book? Or is this just...
No, well, this could be. Color plate 10.4.
Are these the pictures you took out of it for your coloring book?
There are color plates at the back somewhere, or some damn place in here. There are color plates. I don't know where they are. It might be some color plates from there. I'm not sure. They like to clip things. Yes, I think they are. Okay, I can show you. Let's see what you were talking about. Number one was 73. That was the. Where was he? And I got to put glasses on. I cannot see. The Gayu. Let's see. Yeah, stop this screen. This is a pretty damn good book, actually. You should get a copy of it. Let's see. Where is Vertebrate Paleontology? I lost it. I'll be damned. It's gone now. Interesting. She disappeared. Hang on a second. I'll go pull it back up. And that is one way to find it. Oh, wait a minute. No, I'm looking at the color plates. I was looking at Vertebrate Paleontology. What the hell did I do? Oh, it's a Mac. That was color plates from Vertebrate Paleontology. So those were. Okay, so I wanted to go up to, so these are all, come on. Stop laughing at me.
No, I'm not laughing at you. Did you see Dan's comment?
Oh, no. What?
It just cracked me up.
Y'all talk big game people who can't do Punnett squares to that one. Oh, God. And Raw Matt came back yesterday and he talked about his goddamn paper again. It's
I saw that video. I reposted it on Peaceful Science.
Okay, when you get horribly embarrassed and exposed, you wait for what, a month and a half, and then you talk about your paper again?
Oh my gosh. Yeah, and he still hasn't fixed it. That's the thing I was most amazed about. Like, he repeated the claim explicitly in the video that the mother's mutation rate is 0.83 like per year after whatever the 21 was.
Right. He hasn't
realized that R is a correlation factor. R talks about how well the mother's age correlated with the father's age.
Yeah.
That's it. That's all it meant. It had nothing to do with like the mutation rate. They say that in the next sentence. If you actually read the paper, in the next sentence, it says something like, the father's age continues to affect mutation rates. P equals like 3 times 10 to the negative 8th. But the mother's age seemingly does not affect the mutation rate. P equals like 0.49. So it's like not statistically significant in the inherited mutations. Or not mutation rate, but like the number of mutations inherited. I don't get why he's still using that claim. Like, I honestly don't know. I'm at a loss for that one.
Yeah,
that was like the first time I saw that, I had to bang my head against a wall to be able to comprehend. Lose a couple of brain cells for that one. No, I don't know. It doesn't make sense.
That paper has resurfaced. I don't know if anybody can read this. This is kind of weak on here, but this is the Guyo?
Yeah.
Guyo, yeah. And there's the actual fossil.
It's
and
an
then
incredible
and where you ended up was on page 108.
Yeah.
Let me go down.
Oh, I was giving a very brief overview.
Yep. Page 108.
I basically just talked about the fossils and their synapomorphies. I didn't talk too much about like the actual skeletal features and all that kind of stuff.
Yes, you were talking about this guy, Seymoria.
Yeah.
Seymoria. That's what I need you guys for. Teach me how to say shit.
Well, like I said, most of the clade names don't mean anything. Like the clade names, they're just so that people in a paper know what the other people in a paper are talking about.
Yep.
You could say it as long as you're pronouncing the letters. I think you're good.
Diadectes, skull and lateral. That's pretty detailed. Bony skulls and like something I spotted. What the hell was it? Skull diagrams are kind of interesting. And I'm trying to think where did I have those? Hang on a second. These, oh God, where did I see them? These sort of really, what do you call them? Cartoon-like skull diagrams with diapsids and synapsids and whatever. I spotted them somewhere.
Diapsids.
Yeah, but these skull diagrams, I tried to look it up to see if there was some collection of them. Because, let's see, list of synapsids. So where are we here, synapsid? I'm trying to figure out where I saw that. Yes, that's where I saw it. Okay. So actually, I could do this. See if you can see what I'm talking about. Because there is a convention here. And come on, don't do that to me, Mac. And I put it in a different window. And I don't know why I did that. But, okay, so there's that. Our screen, application window. What? It doesn't even have it. This sucks.
Wait, what are you looking for?
I just snapped the picture and it's giving me crap here. Not showing it to me. Hold on. Production values are us. We good at this shit, man. Where are you? Cancel? I'm at StreamYard is not showing it to me, and I'm not sure why. Application window. Oh, there it is. Okay. Share. So this.
What a beautiful diapsid skull.
Yeah.
That's synapsid.
Yeah. This is sort of a cartoon version of a lot of skulls that you see. Is there some convention somewhere where we can learn about these things?
Like synapsid versus diapsid?
Well, the thing about this one is POJ.
Oh, like what all the different like fused bones and stuff.
What do they mean? Yeah, and
yeah, those are just different bones. Mammals have most of those bones fused because our skulls are very fusiform compared to most other lineages.
Right. Where do you find the key for these things? And is there a book about skulls? where we can learn more about.
There's definitely a book about skulls. I don't know where it is, but
we will find that book because that's very important. But that's very, very interesting looking at how all the different skulls came together and morphed, and then trying to find the embryology of this. We know I've got a book on human embryology, and I think it gets fairly detailed about skull development. That would be interesting to compare with other tetrapods. So
yeah, Evo Devo is a really cool field that I should learn more about, but it also makes me kind of sad because you have to kill like baby possums and shit.
You do?
Well, I mean, like, yeah, if you want to discover like skull development, one of the most common, like, what is it called? The animals that scientists use. The name is blinking from my mind, but a model organism, right? One of the most common model organisms for Evo Devo, especially development of like mammalian, bone structure and stuff. Well, for mammals, it would probably be like the lesser short-tailed possum, because marsupials are very easy to cull. So the short-tailed possums are really small, and then since they're born so early, you don't have to like take the embryo out of the mother or something, you know?
Yeah.
You just kill the baby, and it's very sad, but useful.
So Cordova says that that's a feature that God created for us so that we didn't have to kill humans to study us. He created little things like possums and mice and dogs and cats.
Mm-hmm.
You've heard his theory, right?
Sorry, whose theory?
Cell Cordova.
Oh, wait. His theory referring to what? I've heard his stuff on fitness, but I haven't heard much else from him.
Okay, his theory is that God created chimpanzees to be very much like us so that we could kill them to study them for medicine, so that we didn't have to kill humans.
A little morbid.
Yeah.
Well, what about gorillas? Are we supposed to be killing them too, but like a little bit less than chimpanzees?
I don't know. Possums, we can kill a little bit more. Zebrafish, who the fuck cares about zebrafish?
Pennies on the dime.
Drosophila.
Genocidal.
Yeah.
Honestly, yeah, science is very genocidal towards Drosophila.
Oh my God. But, you know, they're not like us, so it's kind of racist. Drosophila racism is a real fucking problem in this country.
Mm-hmm.
Yep. Sticking to that. Anyway, let's see, what was I going to say? See, zebrafish are a model organism that we've studied just extensively, and that's why they didn't make the discovery about paddlefish until about 2012, that they had fingers. They had the finger Hox genes.
Well, and also another problem with paddlefish is that they're extremely rare. You know, there's one living species of paddlefish. The Chinese paddlefish was declared extinct like this year, and the American paddlefish is endangered, so... Really? You know, it's kind of hard. And also, they're very big fish. They're closely related to sturgeons. And they have a pretty long life cycle. So you can't breed them super easily for research. Like, you know, Danos, which is, you know, zebrafish. Danos, their whole life cycle is like a couple of weeks, which is, you know, you can run multiple experiments within a couple of months, right? You can't do that with paddlefish or sturgeons. You probably do with Beeshear. Beeshear is it's a sister group to paddlefish and sturgeons. And they're the only ray-finned fish that still have lungs. So that's kind of interesting.
Here's the paddlefish paper.
Have you seen...
2007. Sorry.
Have you seen the Beeshear paper about phenotypic plasticity, where like the Beeshears that were raised on land developed denser bones and like stronger muscles for land propulsion? No. It's a very interesting one. Let me see if I can pull it up. And I'll post it in the live chat so that you can like do whatever you want with it. Yeah, phenotypic plasticity is another very interesting topic that, I don't know, I feel like there's a lot of really cool evolutionary stuff that I wish I could talk about, but they're not super relevant to this whole discussion. Hello? Is anyone here? Okay. Oh, Speed, you still here, man?
Apparently Speed went somewhere.
Somehow
fast.
I muted myself.
Oh.
I'm mutable.
Shame on speed.
I am mutable. Anyway, I found that paper and I'm putting it. How long have I been muted?
Not
Have I
super
been
long.
muted this whole time?
Like 10 seconds.
Oh, okay. So anyway, I posted the name of the paper by Shubin on the Hawks Genes.
Oh, that was by Neil Shubin? It makes sense.
Yeah. Neil Shubin, Davis, and Don. And there it is.
Yeah. I don't know. I think if I could get any pet fish, I would either get a bichir or a mud skipper. The only problem is I am too lazy to maintain an aquarium. Especially in college. It's far too time consuming.
Three-dimensional limb joint mobility. Let's see here. Looking at that one.
I don't know. I can't find the specific paper I'm thinking of, but I don't know. Like I said, scientists have done studies on bichirs where they raised them out of water and like they didn't ever let them swim. They just raised them on land because they're obligate air breathers as well. Like they have to breathe air. They don't breathe water, and they have a lung. So all they had to do was just make sure that their enclosure was just very well misted, and the bichirs actually grew like much stronger muscles and much denser bones, and their limbs were thicker than like the wild type bichirs.
And you can't find the particular paper? Can you type in?
So the only problem.
Huh?
So the only problem is that there's a bunch of papers coming up. I thought there was only one paper about like phenotypic plasticity and bichirs, but
can you spell the name of that?
Bichir phenotypic.
In chat.
Something will probably come up if you just like copy and paste that into Google.
Oh, okay. I have seen this paper somewhere. I bet I have it.
Yeah, there's videos of them as well, like Bichir on land, where like from the study, I think.
You link that paper to me at some point?
Yeah, if I find it.
Okay, it's mentioned in Rocks Were There. Interesting.
Oh, is it?
Yeah.
I haven't gotten to that part of Rocks Were There yet.
I've got a, let's see. Oh, it's mentioned actually in vertebrate paleontology. Mm-hmm. And let's see, we have comparative phylogenetic analysis of teleos fish Hawks genes in cichlid fishes. Am I getting...
So cichlids, I'm fairly confident cichlids are also a type of labyrinth fish, which means that they can breathe air, but it's not homologous to our lungs.
Okay. Nucleoproteinomic system. We have Wagner, tetrapod limb, a hypothesis on its origin. That mentions Bichir. All these things do. So that's all I can find. I guess I don't have a paper on it, though.
Oh, wait. No. So like gouramis are what I'm thinking of. They're called labyrinth fish, and they have a labyrinth organ, which makes them breathe air.
Yeah. So my one of our employees for a consulting company, his name is Paddlefish. His handle is Paddlefish because he's a molecular biologist turned programmer. Mm-hmm. He worked at the University of Minnesota on paddlefish because we have a lot of them here. So paddlefish in Minnesota, well, they're not extremely common, but they grow quite large. And he worked on paddlefish for years, and he got tired of it because lab work is apparently pretty boring after a while. When you get down deep into one particular thing that you're studying, you don't learn anything else. Yeah, so he knows a lot about paddlefish. He was a lab rat. That's what I call lab rats, people that just study one particular thing all their lives,
Mm-hmm. Yeah. Wait, so he studied paddlefish and stuff. I've definitely talked about this paper before, but there's that one paper that showed the paddlefish sturgeon cross, which is absolutely insane, like a hybridization event.
Really?
Because nothing about them should have been able to hybridize, but they were able to.
Yeah, because we have sturgeons too here.
I'm honestly really surprised that none of the creationist organizations took it and ran. Like, that paper should have been plastered all over AIG and stuff. But I think it's just because it's a fish, no one cares.
Oh, it will be now if Standing is still here.
I've directly talked to Standing about that paper. So I know for a fact he knows about it, but I don't think he ever really mentioned it except for briefly in a live stream after I talked to him about it. But like sturgeons and paddlefish, their ploidy counts were completely different. Like one of them was triploid, but like facultatively diploid, and the other one was like pentaploid, but like facultatively quadruploid, I guess. So their chromosome counts don't line up at all, and obviously like the haploid genomes just didn't have even similar numbers. Their divergence estimate was like back in the middle Jurassic, so they were as closely related to each other as like birds are to T-Rex, or like we are to monotremes, right? Imagine having an offspring with a platypus. That's how absurd that is. It's fucking crazy.
So was this a paper? Amphibious fishes, evolution?
It could be.
Okay.
I think I scrolled through that one, but I didn't see the figures. I didn't have time to read it because we're on a live stream. There's some cool stuff, though. Yeah.
Yep. Let's see. Amphibious. Okay.
There's a lot of amphibious fish, which is why I don't get why people. I've heard Neff say this before, where he said like, "Oh, what selection pressure would make a fish want to breathe air?" And it's like, well, first of all, they don't want to breathe air. It's just it's a beneficial trait. But second of all, like breathing air is a very beneficial trait, especially if you live in like oxygen poor waters, or if you live in a environment that undergoes a lot of change, right? Like seasonal change. Like you want to be able to breathe air so that you don't die. And, it's evolved a bunch of different times, like in different fish lineages, right? So, you have the true lungs from beach ears and lungfish, but you also have like the other completely innovative systems that have evolved from like mud skippers and snakeheads and labyrinth fish and all that kind of stuff, and like walking catfish.
Yeah, and then when you look back into arthropods and how they basically started using their muscles to force fluid around the body, and those muscles eventually became more specialized and became a heart, like a single chamber heart, and then the body cavities became specialized. And those eventually turned into branching veins and capillaries and all of that. And you can see the development of the circulatory system, which is remarkable in itself.
Yeah, it's really interesting. Oh, it goes back to the whole evolutionary.
Nestle has a question. Nestle has a question. Could this be a case for genetic assimilation? So I'm not sure. I think we'd have to roll back to where we were talking about something. Genetic assimilation.
Is he talking about? I'm trying to think of what. Would it be like? Is he talking about the environmental effects on the beach ears? Probably.
I think so. Genetic assimilation. I'm not sure.
I mean, so. What would be the difference between genetic assimilation and phenotypic plasticity?
At this point, I just let any old person in here.
Yeah, go for it.
So, Nestle, you want to stop in? I gave you a link. Or anybody who wants to stop in.
I would also like to preface that I know basically nothing about this shit. Like,
yeah, he said plasticity of the Bisher.
Yeah.
We will look into it.
Yeah, I have no idea. Yep. I feel like there's two people in the chat that would likely give a much better explanation of what that means.
There's just a tremendous amount of work, and I have to focus more because fins to limbs is one piece of work I really want to do, and in Evo Devo, and get down to the point where I can explain these morphogenetic fields or morphogenic fields that develop that. They happen very early. I mean, coming out of the mother, the zygote has basically a posterior anterior field set up in it, and so that's just in the zygote. And from that, everything starts to it convolutes and involutes and develops and creates all of these different, like if you imagine a river flowing. Like if you were to drop water on a pile of dirt, it would form rivulets, that sort of thing. And each one is a particular tissue type, and it keeps going down and branching. And it's just an amazing way to look at evolution. And it is the most convincing thing about evolution when you look at Evo Devo. Mm-hmm. So, but I'd like to know what I'm talking about at great detail before I try to convert it into something layman-like. So I'm working on it. So
yeah. That's why I don't know. A lot of these things, like I know about, but I don't know it, one, to any great extent, and two, enough to like explain it to someone else, you know?
Kind of a continual problem for everybody, you know?
Yeah. Like I don't know. I feel like I can list off fossils and like the current understanding of like cladistics pretty well, but it doesn't mean that I have a total grasp on how those like lineages are derived, you know?
Right. Yeah. Same.
Yeah. What do we have here? Therapsida, late Triassic. Now I'm looking at my vertebrate book, I believe. I'm going to have to hook you up
with the book.
Yeah.
Through totally legal methods.
Totally legal.
Yeah.
Well, you know, I think I'm going to say this publicly. Immutable Destiny whined for 6 months until I bought him a glycobiology book, and it wasn't cheap.
Mm-hmm.
It was not cheap. He whined and whined. Do you remember, Andrew, how he whined?
I remember how he whined. I definitely remember this.
Andrew remembers because he was he basically had a part in the whininess of Immutable.
Immutable was using him as like a bargaining chip almost.
He was, yes. But he finally got his goddamn glycobiology book, so now he's happy.
For respect of my friend, I'm not going to say anything anymore.
Yeah. Immutable's not here. We can trash the shit out of him.
Yeah.
Like, I don't know.
That's what Friday nights of the Atheists are for. Trashing each other.
Yeah, he hasn't even shown up today. Usually he shows up. But anyway, this was pretty cool. I think you want to put a stopper in this?
Yeah, I mean, I don't. If you want to talk about other stuff, I can talk about, at least to my understanding of like paleogenomics of like merit ungulates. That's something I've been looking up recently. But no, yeah, I that's roughly the extent of my knowledge. I could have a bit of a conversation like going further, but
yeah,
that's about it.
Well, we'll keep going. Maybe we'll find when we share this book, we'll find more stuff in there to discuss. And I got a couple other books on fins to limbs, but I don't know where the hell they are. They're over here somewhere. But we'll find those and get some more pictures and maybe go a little bit further into this at some point.
Yeah. Like I said, it's fun talking about stuff that's not directly related to creationists. It's a good break. It's a little breather there.
It's fun to talk about the actual science, you know. Yeah,
because like I was talking to Erica about this a while ago, and she says like her brain has two modes, and it's like actual science mode where it all the scientists are like consistent with reality, and then there's the creationist debunker mode where you have to question like every single sentence.
Oh my God. StreamYard is not even letting me post things. Anyway. I'm always blowing people off when they invite me into a stream at like 12 a.m. or 11 p.m. or whatever. It's like,
is that referring to Standing for Truth?
Him too, yeah.
And I mean, like, all the streams are like 3 a.m.
Huh?
All the streams are like 3 a.m.
I know. It's like, Jesus Christ, my brain is like, what? I have 10% of my brain by that time at night, you know? I mean, I'm ready to go watch TV. And these guys, they just get rolling at about, 11 a.m. or 12 p.m.
Yeah.
Or 12 a.m.
I don't know. There's no way I could do that. And I have a pretty flexible schedule as like a college student with most of my classes online. I could sleep until like 1 or 2 every day if I wanted to. But like, 3 a.m. live stream for like 2 hours is just, it's a little much.
Yeah, it's just that I'm incoherent at that point. If I've been up.
Dude, I'm incoherent at 5 p.m. in the afternoon.
Right, exactly. Yeah. I am coherent at about maybe from 4 a.m. to about 9 a.m. and that's about it. The rest of it is just
I've realized that I'm only ever productive in daylight hours, right? As soon as the sun sets, like I'm fucked. I can't do anything.
Oh yeah. Yeah, the old sundown depression. That's God, in Minnesota in the winter, it's just a continual problem. So I've got I got these bright white lights, they're called the pure white light. And I have I got 17 of them in my room, 100-watt equivalent.
Yeah.
They're fluorescent. I have to do that. I have to blast myself with that constantly in order to survive a winter.
Yeah, I don't know if that would work for me. I might just have to get like a fusion reactor to emulate the sun.
Oh, that works. Yeah, fusion reactor. Very good.
Why don't you just get a blue dwarf star or something?
Just keep it in my room.
I am trying to get a hydrogen bomb for 4th of July.
And you are now on the FBI watch list, my guy.
Oh, shit. I said that out loud?
Not out loud. You said it on stream. Either the creationists are reporting you right now, or the FBI, like whoever the dude is that's been ghosting us for a while. He's like, all right, guys, we got him.
But my neighbors are popping off. Like they go out and spend like $5,000 on fireworks, and I'm like, okay, dude, look what I got. 10 megatons.
And just like that, all of Minneapolis is gone.
You want to see some fireworks? I got some fireworks.
Yeah, you'd be going out with a bang.
Yeah, going out with a bang.
You want to see some fireworks? Go to the sun.
Actually, there's two ways that I would end my life if I had like a terminal disease and I had to do that. One of them is in a Bugatti at 280 miles an hour, hitting a brick wall or a rock wall. And the other one is strapping myself to a hydrogen bomb.
Yeah, I've sort of thought about that too. I want to go out like in a cool way, and I want to have control over like how I do it, right? I would love to be like strapped to a bomb and just, fall to the earth or like get shot out of a cannon or something. It'd be pretty sick.
Dr. Strangelove, Slim Pickens rides the bomb down in the end.
Yeah, like that.
Yeah.
Jump into Mount Vesuvius.
Oh yes, that's another
jumping into a volcano. I don't know about that. I would want to do something that's pretty like instant, like even if it would be pretty badass to like drown or like get eaten by a shark or something. Like, I don't know if that's how I want to go.
Yeah,
I want my last seconds to be like I'm a badass, not like I'm in so much pain. Please help me.
What if two volcanologists walk up to each other and one says, "Do you know the chemical structure of lava?" And the other volcanologist says, "Okay, let's go find out." Oh, he's dead.
Yeah. Like I described how I want to end my life. Reading and studying like as fast as I can. It's like I don't want to get to the end of the dock and start to slow down and kind of fall off the end. I want to run off the goddamn dock at full speed.
Yeah.
So, and I know this neuroscientist. He's like 92. Joaquin Fuster. He knows more about the human frontal cortex than any other man alive, and he is still going, strong, which is really cool.
Yeah,
I mean, that would be the ideal way to die, is to be running as fast as you can off the end of the dock. So
it's pretty crazy how many professors like teach until the day they die.
I know there was this one neuroscientist, I can't remember his name. He was like biking to work at like 101 or something like that, or biking to the lab.
Oh my god!
Like Jesus Christ, that is awesome.
It's the opposite of the oldest doctor alive Onion video.
Oh, I haven't seen that one. I love the Onion. Okay, we got to end this thing. We got to shoot this thing in the head. Okay, guys, everybody in chat, thank you for showing up, and hit the goddamn like button, because every now and then the creationists stop over and they hit my unlike button, and it's starting to piss me off.
So there was a few of them in today's chat. None of them hit it. So
no, they didn't. And I'm really proud of Stanley that he didn't hit the dislike button. So,
unless he's still here, now he knows.
Now we know.
Please, everyone in the chat, subscribe for Mr. Speed.
Yes, because I'm going to die soon. Okay, ending the broadcast. We'll see everyone. Bye bye.